Hello dear viewers, myself Nasir and you are watching Mr.Zoologist YouTube channel. Today we are going to discuss about DNA double helical model that was proposed by Francis Crick and Watson. So we are going to look upon DNA structure. In actual, what is DNA? You have to first of all understand that DNA is an acronym. It is an acronym of deoxyribonucleic acid. It is an acronym of deoxyribonucleic acid. First of all, Frederick Sanger described or discovered them nucleic acids from the pus cells. He discovered them from the pus cells. They were present inside the nucleus of the pus cells. That is, and their nature was acidic. That is why he called them as nucleic acids. So that was the discovery of DNA and RNA. They were both of the class of nucleic acids. DNA is a double helical model, whereas RNA is a single helical model. There are two types of the nucleic acids. One is RNA and the other is DNA. Ribonucleic acid and deoxyribonucleic acid. So from the X-ray studies, Watson and Crick proposed DNA double helical model. As you can see from here, that is one helix. That is one helix and that is the other helix that wounds around the first helix. If you say that is a 3' end, that will run from here and there from 5'. As well as you can say that if that is 5', then that side will be 3'. These are the two strands which runs in a parallel direction, and these strands are joined by nitrogenous bases. What are nitrogenous bases? And we will discuss them later now in this chapter, in this topic. That is a double helical structure of DNA. If it contains double helix, then that will be double helical DNA structure. And if it contains single helix, that will be the RNA, ribonucleic acid, that is single stranded. And if you are going to look at the composition of the nucleotides, these DNA are made up of nucleotides. And these nucleotides are composed of 3 components. How many components do have a DNA? DNA nucleotide contains 3 components. These components include phosphate group which is attached to the nitrogenous bases. One is phosphate group, on the second is nitrogenous bases which are used for the attachment of the strand. Nitrogenous bases, and the third one is sugar. These two components are similar, and there is just one difference of one base in the RNA as compared to those of DNA. That is, in place of thymine there comes uracil in RNA, and these bases will represent the bonding between the nucleotides. I will show you that how this bonding occurs, hydrogen bonds, how these bonds are formed between the two strands, and bases join them together. And sugar, if we talk about DNA sugar, that is deoxyribose sugar. If we talk about RNA sugar, that is ribose sugar present in the RNA molecule, single helix. And these helices are joined by, there are 4 types of bases: adenine, thymine, guanine and cytosine. These 4 nitrogenous bases are present in DNA, and these nitrogenous bases are always in the composition in the fixed ratio attached to each other. Adenine always forms pair with thymine, guanine always forms pair with cytosine. That pair will always be similar. You will never see in DNA structure that adenine is bonded to guanine or either adenine is bonded to cytosine. You will not ever see that type of interaction in DNA helix. Adenine will always form double hydrogen bond with thymine and guanine will always form triple hydrogen bond with cytosine. As you can see from the hair structure, adenine and thymine will form double hydrogen bond with thymine, as well as guanine will form triple hydrogen bond with cytosine. And these type of interactions run throughout the nucleotide of the DNA. That was the structure proposed by Francis Watson and Crick, that DNA is a double helix, that one helix runs from 3 prime to 5 prime and the other runs from 5 prime to 3 prime. That is the DNA double helical model, and these are the bases that are used to attach the two nucleotides together. And one nucleotide is composed of 3 components. One is phosphate group, the other is nitrogenous bases, and the third one is sugar. Sugar is of different nature in DNA and RNA. That was the topic of today. Hope so you enjoyed it. We will meet you in the next video. Till then, peace out.